Yeah, so I'd like to talk about today, well, one way of expressing these things is in terms of supertypes. Supertypes of a certain class of object called alleles. So this would be of alleles, and that's associated to this group of alleles we write sometimes MHC2. Supertypes are going to be given more in terms of a mathematical setting. A bit of biology, not too much, and more of a formal mathematical development. On the other hand, we have the notion of a serotype. Not to be confused too much. And this is a classification or some kind of a structure on the space of the same set of alleles. And this is given by the World Health Organization. They classify the serotypes, associate serotype classes to a certain number of alleles in the same body of alleles. And those are done traditionally very much by medical laboratory work. And in fact, even fairly recently, they give credit mainly to a bunch of laboratories, bone marrow labs, many, many different medical laboratories and groups of laboratories, International Cell Exchange, I think. And they can all be read off on a very nice table, which we use. There's a table of all of these alleles together with what they found by the WHO. That's something that's almost like our Bible. There's a list of about 800 plus or minus alleles that we're dealing with in this class. Okay, so what we want to do here is to relate these more mathematical objects with the medical objects. That's the goal of today's talk.
You mean to insert that 800 in the supertypes or in the serotypes?
That's alleles of 800 alleles.
In either case.
Yeah, I mean, these are the same.
All those are the same.
Yeah, yeah, those are the same. Okay, so that's the goal of what I want to do today. And just give for a frame of reference, I could write down, I think, this was from last time. So this is an example. These are examples of alleles. I think there's 12 or 14 of these in this column. And these are the way the alleles are named in this big database of approximately 800 alleles. It changes from year to year. They add new alleles, but the one I'm speaking of is a list of around 2008, which has got more data, because some of the more recent alleles that have been discovered, they don't have much data. So that's why I like this database. You can find most of these things I'm saying on the paper we finished yesterday, day before, on the website for the conference. That's a good reference for this talk. And it has got 40 or so papers, and it lists the databases, the reports of WHO, and so on. So that's a good reference for these things I'm saying. Okay, so here we have some alleles. We just call these alleles. These are the coding for the alleles that everybody understands. I mentioned yesterday, before the star is a gene. There aren't very many genes in these classes. Maybe just 5 or so, 4 or 5. There are few genes. But the genes have representations as alleles, depending on the individual. So a gene makes an appearance, maybe 2, in individuals. And that's the allele. So the allele is an appearance or a representation or a gene expression. And so we're dealing with these alleles, and the gene part you can read off from the code by the first symbols before the star. Okay, so let me just recall again. The second two here, after the gene, represents a family. Here's just a family here. Here for DRB1 star, there's a number of families, as you will see. And the WHO, more recently, they say these families are associated, when possible, to a serotype, to disease. Now, a serotype is a genetic version of disease, but it doesn't exactly define a disease. It's a very rough, you might say, it represents a weighted class of, small weighted class of diseases or various aspects. And these alleles represent responses to the same diseases. So the alleles are associated with a certain pretty well understood process of an immune reaction to a disease or different diseases, or more generally to invaders into the human body, viruses, bacteria, even cancer. Okay, so that's the idea, and we're going to be working with these. Coded, we're using the same code as I have in this list. The code changes too often, but that's the one we'll be using. And so if we look at, so these are the alleles with the most data. Here we have data relative to this number of peptides. Those are alleles which are understood best from the point of view of medical experiments. So these are the ones that have the data. Here we have the number of peptides which we know the binding to this allele. There are 924 which have data which tells how much these 924, each of these peptides binds to this allele. Okay, for today we're not talking about peptide binding, but I just put this up here so we can see what we're starting with. We're starting with these alleles, but there are many, many more of them, same type of coding, and there are going to be about 800 alleles in the databases that we're working with, about 800. And so what we want to do is start with these alleles. So we get the database of the alleles from as closely as possible to sequencing experiments. Experiments which go from the double helix through some kind of genetic code via RNA and eventually end up as an amino acid sequence. So the most common representation of an allele like this is an amino acid sequence, which we've studied in the first two lectures. So we'll be looking at amino acid sequences of these alleles. Okay, so we have to do, we don't want to mess around with the what we get from sequencing very much, but we do just a little bit to get a starting point, and we want to cut away parts of these amino acid sequences which are not very important for that allele in terms of immunological response. So we end up, each of these alleles they're given to us with a position, and we're looking at positions, I think it's 9 to 89. So these are the positions in a longer amino acid sequence. So here we have part of the chromosome, an exon. In this case, it's exon 2, and it gets processed by these genetic codes, etc. And it gets a little loose around the end, so we have to be pretty careful to start this at the right place. So, of course, from the micro probe, we're going to get some kind of a band across here. We want to start this at the right place, and we have enough information from the databases to start this at position 9. That seems to be about the right position to catch the important positions and residues in this amino acid sequence. Stopping here, also okay. In general, most of the alleles are presented from the laboratories in sequences, eventually amino acid sequences, which are somewhat longer than 81. These are 81 length. So they're presented, come out of the laboratory processing in sequences. Some could be 150 positions in the sequence, or maybe 200, and occasionally shorter. Sometimes they don't even reach 81 positions. In any case, we start here. We start with these sequences. By now, they have become amino acid chains, and we have a first position and a last position. Maybe some of them are sparse, a bit sparse. Few of them are sparse. Okay, so that's going to give us some kind of basic space. I'm going to call that space script n. This is, well, it's not a supertype. No, it means that there's some gaps in it. Yeah, it could be gaps in it, yeah. Yeah. So this space I'll call, all along here is the space script n. So it's a set. And also we do a couple of things. If two alleles have the same amino acid chain, we only count that once. And we use the amino acid which comes first in the WHO list to represent that. Okay, so by the time we do these things, there's some that are called undefined. We exclude a few undefined ones because even in general, many of these are not very well defined. So when the WHO says undefined, we exclude that. And then we end up with about 785. This is the number of alleles in this set. So it's a finite set consisting of about 785 amino acid chains representing the same number of alleles of this immunologically generated genetic base. Okay? So then what we want to do is to put a geometry on the space. So we want a geometry, what I would call before like a Euclidean geometry on the space n. Okay, so it's a finite space, so we can't use Euclidean geometry per se, but we have described kernels k hat 3, k3, k hat 3 on amino acid chains. We can restrict that to the space n. So we have a kernel on n from previous lectures. It's a kernel on amino acid chains, which is given, well, by these K1, K2, K3 via some version of local alignment. Okay, so this is a geometry on n, but it's more literally a geometry when we define a distance function. I want to define a metric space structure on n. So this will be, we'll see here, the geometry given directly in terms of just a metric space. It's a metric space structure on a finite set. Okay? So the way we do that, in fact, if we have any kernel, suppose any kernel k on x, on any set x, let's say x finite for here, we can define a distance, the distance from A to B, A and B elements of X. We can do this in two different ways, both of which are useful. The first distance function is the following. Remember, k sub x is the representation as a real valued function on x. It's a map from x to r. And k sub x of y equals simply kx y. Okay, so we can represent x as real valued functions on the space x. Okay, so now for a distance function on x, we can do the following. We can just take the norm of ka minus kb in this inner product space norm. The inner product space norm k it's a norm on the space of functions. It's given to us, as I described before. It's the inner product on the space of functions on x. And that's what this means. If you have a space of functions on x with this norm, then you have a natural inner product k, then you have this natural norm defined, and that's a distance. We call it the kernel distance function. It's a good notion of a distance function. Okay, but there's another notion of a distance too. We can call this dL2. I guess, does that read it? Maybe I should write it here. D, I call L2 of A and B. There are two notions of a distance function. This is for any kernel, any time. Okay, so dL2 is simply the sum of k a of t minus k b of t. And here we take this inner product, square it, then we write square here, and then we divide by the number of points in x, and then the sum is over all t and x. This is another notion of distance. And they're related because this could be interpreted as a random walk on x relative to this distance or this kernel with two steps. This would correspond to one step. So it's a little bit it is like taking this random walk. And it seems to be a useful construction. We call it the L2 distance because we could think of Ka and Kb as elements of this space L2 with uniform measure on the space x. Then we're just taking the L2 distance. Okay, so that's an important notion for us, is this L2 distance. Maybe more so than the kernel distance here. Actually, they are related somewhat by the covariance operator, but let me not go into that. Okay, any questions? Okay. So what we want to do is the following. I want to notice the following. From the serotype studies, the World Health Organization, they name a lot of serotypes, and they have a lot of these numbers which are like serotypes. These, like here we have a 13. That's a little bit like serotype DR13. These are, not exactly classified into serotypes, but these codes are associated to a serotype in some way that the World Health Organization tries to use when they guess about something about medical properties about the allele. And sometimes they officially assign a serotype to an allele. So you read this WHO literature and you'll see how they are a little vague about the importance of the serotype classification, but also do a lot of effort to try to put an allele into a serotype, and sometimes they assert they have done it. So, okay, but there are two of these. These are these serotype classifications, DR11 and 13. Two of them, here's a 13 right here. This is an allele in that class. Okay, and there's 11 here too, this one. Okay, 11 has been a big, these two, and maybe two or three others even, but these two in particular have been a big challenge to the researchers of WHO, the people who work on those committee making assignments. And they say that in the case of these 11s and 13s, they're less sure, and they sometimes talk about a continuum of classifications. So they do say that things become uncertain in serotype classification, but especially 11, 13, and two others, 12 and 14. Okay, so when we work on these things, we are also making classifications like these supertypes I'm going to talk about. We have trouble too. We had trouble for quite a while with 11, 13, and the other two that the World Health Organization had trouble with 12 and 14. Eventually, we got 12 and 14 under control, but we never did get 11 and 13 under control. And with many, many experiments we did from different points of view, we concluded with some confidence that these don't cluster. That's what we believe. And it's possible that you have the sets which don't cluster. In my example is you take a circle, and now you take a uniform set of points on here, so it's finite. How do you cluster that? You don't cluster it. There's no good way of clustering that. If you take the points on a circle, take a finite set, uniformly distributed finite set of points in it, there's no way of making a good cluster. You could say, well, let's cluster it. Here's one cluster and here's the other, but you could just move this anywhere around and it would make just as much sense. But also, we found from our experiments that this doesn't cluster. These two, the union of these two, and what you'll see is something that suggests this picture, and that's why we started to do the following. Clustering, which I'll speak mostly about now, corresponds in topology to the number of connected components of a space. Here we're using appropriate topology for finite sets. But so that's the zero-dimensional Betti number. So what we're doing is now, for the union of these two sets of alleles, we're looking at the first Betti number, which I'm not going into here, and we don't know. We've proved that at certain interesting scales, this is non-trivial, like in the circle. So we want to pursue that more and more, and to see if one can find some medical meaning for that, which we have not done. In any case, what we're doing now is to exclude these because they don't fit by what we do and by what the World Health Organization does. So we're looking at a new space, M equals N, the space of all these alleles, less DR11, 13. Yeah, so we're going to look at this new subspace of alleles where we've excluded these two subsets, subsets 11 and 13. So we're looking at the remainder, and here we have, I think it's about 450. So M equal 500, something like that. Maybe a little more or less. So we're going to look at that space. Okay. And so for that space, I want to do what we call a cluster tree, because we're going to be looking for the structure of supertypes. Supertypes in that set of medically important alleles. So on the space M, we have a distance function now. The distance function here is simply dL2 restricted to M. That's given as a distance function on N, which contains M, so we can restrict it to here. So here we have a distance function given on a finite set. Okay, so the clustering theory is a very big deal. And everywhere you look in these kind of modern statistics and medicine and pattern recognition, intelligence, and so on. So there's been a lot of work on that, and ours overlaps with that, and we think we use that. Okay, so I want to make a cluster tree. It's a bigger object. It contains more structure than just a partition of the space M. So I want to describe how we get this structure things. It gives not only the clusters, it gives the relations between the clusters. So it gives more. And we get the distances between the clusters, the minimum distance between each pair of clusters in our investigation. All right. So towards that end, let me do the following. And it's very much inspired by this linkage clustering. It's called linkage, and usually it's minimum. And I'll describe that thing which is used quite often. It's good for finite sets, not so good when you have an infinite set. What is the minimum linkage? We start with a partition of the space, say M in this case, into singlets, into single alleles. So here we start with our 500 clusters of one each. Okay, next stage, we take the two that are closest, the pair of these, which is the minimum distance, is measured by the minimum distance. That's the minimum linkage. Okay, in general, we want to do this recursively. So say here we have two that are pretty close. So these get clustered together in the first stage. They're the closest. So we put those into one cluster with 2 points. Everything else has 1 point. Okay, now I want to define a tree of clusters in the following way. I just repeat this step at some later stage recursively, where this linkage is given. Up here I have a cluster here and a cluster here and so on. I take the minimum distance between these sets. These will be sets after some sequence of steps like this. We have some sets up here, subsets. It's going to be, we'll have partitions that we could organize these into a partition, and then we'll take, in this set of subsets, we'll take the two subsets which are closest relative to the minimum distance of sets. It's a distance on subsets, not the distance on our space M, but a distance defined by M on subsets. By taking the minimum distance of two clusters or two subsets, we want to minimize that, then we would join that and continue.
Just take the minimum.
What?
If you want to take distance between two clusters, you just take the minimum of all the distances between 2 points.
Yeah, that would do it. That would do it, I think. Yeah. I think that's right.
That's usual. That's
Yeah, I think that's right. Yeah, that's just the minimum of all pairs in the cluster. And we want to do that for each pair of clusters. And then we'll choose the two clusters where that is the minimum, and we join those. The minimum linkage is probably the most common and perhaps one of the best. And you keep doing this, and finally you get a rooted tree. So at the top you get a root and then it comes down and so on. It's kind of binary. You can think of this as a binary tree. Because we can assume that these distances don't coincide. They're real numbers, so they're not likely to coincide. And we assume they don't. And if they did, we could still deal with that. Okay, so there are many ways of replacing minimum linkage, the minimum part, by other notions of distances between two subsets in a partition. These are subsets of a partition. So they're not just ordinary subsets, they don't overlap. That's important. Okay. Okay, let me come down here. So here's the distance function I'm using. On the space n minus, m itself has been endowed with this distance. I wrote here dl2. Now there's a couple of other things we could do, and we do. Replace this notion of a minimum distance, as Mike suggested. We could take the Hausdorff distance at each stage between two clusters. Hausdorff distance has got an advantage. It's actually a distance function on subsets. That's nice. Okay. Or we could take the average over all points. We could take the average over all the xy, where x belongs to cluster 1, y belongs to cluster 2. We take that average of the set of real numbers.
And this will be the distance?
No. No. But actually what we do is a lot of the time we've used the Hausdorff distance, but lately we've been shifting to a new notion closer to this. What we use is called OWA, ordered, weighted, I don't know. What?
Average?
Probably. Ordered, weighted average. And that's not too bad, but here we would put W here, WXY. These are weights. So these weights are characterized by the sum to 1. Okay, now we order them in such a way that as these distances get smaller, these weights get bigger. So they sum to 1, and you see a graph something like this. So the weights are along like this. The weights get bigger and bigger as these distances get smaller. So what one has to do formally is to take all possible distances in that set. We get this unordered set. All these d of xy looking over all pairs. They're all pairs x and y. We just get a set of positive real numbers. Okay, now given any set of positive real numbers, then we could order them in decreasing order. So as they get in this decreasing order, we can now say, use that order to and this graph here to give these weights wxy. I think I've almost given a complete definition. Okay, and that's the main weighting I'm talking about in the supertype classification. I'm using that, OWA. And
if you change the weights, it changes.
Oh yeah.
And do you really see the difference?
Yeah. Oh yeah. We're not the first to do this, but I think, of course, we're the first to do it in this connection, but people have used it, different places. OWA has become a fairly big thing, and the idea is, here's the idea, that if two of these alleles are close, that's much more important fact than the two alleles being far away from each other. So that's why the motivating force behind this mechanism of OWA. Okay, so we're using that distance together with this linkage clustering algorithm. So that's going to give us a tree. It's going to give us a tree in this space of alleles M. Okay, so that's the main object of what I want to discuss today, is the cluster tree on those alleles where alleles MHC2 where I've excluded DR11 and 13. There's still about a dozen DRs or serotypes left. Okay, so that's where we are. And let me just show you, let's see. There it is. That's it. And this is just the top part of the cluster tree. It goes down, down, down until we get singletons everywhere. So this is just the top part of the cluster tree. Okay, so eventually we'll have software that can just click it and you can do like a Google map. So you can go down in this corner over here and so on. So you can go anywhere you want, just like on a Google map. For the moment, we just throw out the bottom part. Top part's the most important. So here we have at the bottom the diameters. So these represent clusters. These 20 clusters. They're all clusters obtained by this algorithm that I just described. Okay? And these are the diameters of the clusters. So here we have a 0. Well, that means it's a cluster of just one allele. 0 diameter. If it had 2 alleles, they're different, so there would be a positive diameter. Okay, and up here we have the diameter of this one, union this one. Across here we get the diameter of the union of the 2 branches before that. It's the same thing. I prefer almost this point 1. We go right up at this bar right here because these vertical lines can be interpreted as higher order clusters, higher order than the bottom ones. And those have diameters, and they're given by these numbers. Okay, is that very clear or confusing? Well, I just wrote it there. But we know from this construction, there's not much things to be proved here. It gives you a tree. It gives you a tree, and each of these branches in the tree corresponds to a subset of the alleles. And that's what we have. Fair enough? Okay. So then the next step is to what does this mean? Okay? And so let me go over to some other slide. Okay, here. Now here's what is contained in all of those clusters at the bottom. These are the contents. I call them bins because they used to be more like a bin. Okay, so these are the bins and the contents of the bins. See at the top we have DRB star, DRB1 star, so it's the first gene. Then we have a 14. So that means it's in this category the World Health Organization thinks it's a 14. And so this first column here, not this one, columns over here, these are the names of the alleles. These are the names of the alleles in cluster 1, cluster 2, cluster 3, cluster 4. And sometimes we have these things here. These are what the laboratories of WHO has convinced them they should assign serotype DR14. So these are the assigned serotypes. Here it means they have not decided because there's too much ambiguity in the lab reports. Here they have not decided. But they have enough evidence they will still call the 14. Because remember, this element of this is the family, they say, if possible, put it in the serotype classification. Otherwise, they seem to want to get it close. Okay, so here we see the contents of the first 4 bins. And you notice what? They're all, I think, maybe they're all, well, here's a DR13, which is a little bit of anomalous because we've thrown out DR13. So I don't worry about that. But the rest of them, look at all these DR14s. This is just another name for DR14. And actually, DR14 is a split serotype of DR6. So these are all DR6s. Okay, so you see that it's pretty solid picture. Includes even these alleles that were found recently. This is an allele which was found, I think, 2010 or 2011. Most of these were found by 2008. So we get some of the new ones, which they don't have much information, just enough to give it a label here. Okay, so it's very clear that, what those clusters, the leaf clusters, look like. And let's just see it next page. Okay, here, we start going, see up here there's a DRB 1*14, which is assigned DR8 by the World Health Organization. So look at these. That cluster 5, everything is an 8 except for these 2. And these are very recently found, and I'd say it's a toss-up whether we are correctly classifying them with the DR8s or whether they should be classified with the DR14s. I don't know. It's probably a touch and go. But otherwise, except for these 2, everything here is exactly DR8 or close to DR8 enough to be put with that label, DR8. Okay, so so here's what we've done. See, we put DR14 over the first 4. And here we have DR8 over the next one because of evidence that we've just seen. So you see what we've done. And then we add on all of these I should call this ST because these are our supertypes. DR14 should be called ST14 because it's our assigned supertype from this information that we've used in this cluster tree. The information being only the sequencing and the blossom information of the alleles. And I still get a little amazed with all the medical information and so on. We're doing the same thing. Every one that's been assigned, we have it assigned the same way. There's this, see, these are the ones that are assigned. Every one that's been assigned, we have exactly the same. Then there are the ones like this. See over here, they haven't assigned them a serotype. So there's a couple here that we can't say that we do that. Yeah?
Those alleles that are in the same class are supposed to have the same binding affinity or something like that?
Well, this is one thing good about this. Nothing to do with binding affinity a priori. This is all pre-binding affinity. This is more directly medical, trying to classify these alleles. First by our algorithms, not using much medicine, and comparing how it compares with the medical classification of the WHO, World Health Organization. So this is pre-peptide. Eventually this will impact peptides, sure, eventually. Because, here I think the main points I want to make, what this does, let me go a little farther in this picture. Here's the 8s, here are the 4s. Look at these 4s. See, they're all exactly the same as the World Health Organization. Here, even the 04s without designation, we get everything the same. Nothing different. And it keeps going on like that. There's a couple of things. Yeah, see, DR2 is the top these top two. Cluster 7, we just group with the previous one to get the 4s. And then we have cluster 9 and 10. That's split. These are split serotypes. DR2 is the serotype, which contains the DR15, DR16. And even the split serotypes, we get pretty much the same. And over here we have cluster 11. This is what are called DR52s. This is a new gene. I mean, another gene. This is, for example, well, that looks still pretty good. Is something missing there? Maybe not. Oh, yeah, here it is. DRB1 star. Not been assigned, and we assign it to a different cluster. Okay, so what's that mean? Well, the thing is, we have other clustering systems, and it always comes out, this goes into this DR52 group. So it's a kind of independent evidence that this should belong here. And the World Health Organization may have slipped when they put a 3 here. I don't know. I don't know. Okay, and that happened once or twice more too. And this is 15s. See the 7s lining up here, the 12s. Oh, here's one. Here's a 8. But that's with an undefined after it. U stands for undefined. So I don't feel too bad about that. When it's undefined, I don't worry too much. And here's a short string. SS stands for short string. Okay, fine, fits in. Okay, well, that's the picture. And to a great extent, what we want to do here is to corroborate our geometry. We've used that geometry on peptide binding on these different experiments I talked about last time. Fixed allele, pan allele. There are framework was level with the state of the art. And so what this does, it gives another confirmation in this, really, I think, extremely surprising way of this geometry from our K3. Okay, well, thanks very much.